Hello, everyone. So Cool Bean asked about how long does the virus survive on different surfaces, which I think is a very important question because, it'd be nice to know how long these things survive. So when we come into contact with surfaces like doors and countertops and stuff, how worried do we need to be and how long does it survive? That was a really important question. I thought so a long time ago. And actually back in February of 2021, I made a video called How Long Does the Virus Survive and How Can You Kill It? However, it was removed by YouTube for violating their terms and policies. It says this video has been removed for inappropriate content. I try to appeal it. I think they like you know, I don't think anyone actually looks at it unless it's like a third or fourth appeal, and you're about to get your channel canceled for inappropriate content. But anyways, for some reason, their computer system, their artificial intelligence, or their deep learning algorithm, their algorithm tagged it as inappropriate. I have no idea why. But anyway, it was a topic that I thought was very important, and I made a video about it almost a year ago. And so I'm going to try to make another video and see if YouTube flags this content. All right. So, people wanted to know how long this virus survives on various surfaces. And in April of 2020, in the New England Journal of Medicine, which is one of the premier journals in medicine, they ran a study. And what they did is they aerosolized the virus SARS-CoV-1, which is just regular SARS, and SARS-CoV-2, which is COVID-19. And they also looked at it, so they looked at it under 5 environmental conditions: aerosol, plastic, stainless steel, copper, and cardboard. Okay, so here is the meat of the question. So what you see here is this is a logarithmic scale. So if you have a linear plot, and these are all linear plots on a logarithmic scale, that means it has a logarithmic decrease in the number of viable viruses. So what they did is they aerosolized it and they looked at it over time, and you can see that the amount of surviving virus decreases over time. And then this is copper, cardboard, stainless steel, and plastic. So what you notice is in copper, the virus dies very quickly. On cardboard, somewhat quickly. Stainless steel lives a pretty long time, and then plastic is the longest. So under the of the 5 conditions they tested, the longest survival was on plastic. And then if you want to look at the way they measure this is by half-lives. And then, so let's see, let's look at this. See, on cardboard, no viable SARS-CoV-2 was measured after 24 hours. Okay, so on cardboard, it looks like about 24 hours. And then, let's see. Okay, the longest viability of both viruses was on stainless steel and plastic. The median half-life of SARS-CoV-2 was approximately 6 hours, 5.6 hours on stainless steel, and 6.8 hours on plastic. So what half-life means is that so for example, on stainless steel, every 5.6 hours, the number of viable viruses decreases by half. So if you started off with 1,000, 5.6 hours later, you're at 500, and then 5.6 hours after that, you're at 250, then 125, and then 67.5. It just drops in half every time. So it's not like it drops by like 500 and the next thing it drops by another 500. It just drops by half. And so that's how most of these virus viability studies, that's how the kinetics of the viral death happens. So the viruses, half of them die, and then half of them die. So it's not like there's a certain time and then everything is gone. It's just at a certain time after multiple half-lives, it gets down so low that's not detectable anymore. And probably there's not enough stuff for it to be causing you any problems. So usually about after like 3 or 4 half-lives, the thing has dropped so far that it doesn't matter. So, 3 or 4 half-lives would be about 24 hours on these cases. And so to be non-detectable, maybe you have to go 5 or 6 half-lives. So for cardboard, it was about 24 hours, and cardboard, if you look at it, cardboard was probably twice as good. So, probably 48 hours for stainless steel or plastic to have completely non-detectable virus. Then this is another study from Lancet, which is another well-respected journal. It's a subjournal of Lancet called Microbe. So stability of SARS-CoV-2 in different environmental conditions. So similar study. And what they did is they measured it at different temperatures, and they were tested by infectivity to make sure that the virus was viable. And they said that the virus is highly stable at 4 degrees. So that's in refrigerator temperatures. So as temperature goes up, so at like room temperature, it's less stable, so it dies faster. And then we'll talk about what things make the virus die faster. So here they go. Here, let's read this part. We further investigated the stability of the virus on different surfaces. Briefly, a 5 microliter droplet of virus culture was pipetted onto a surface and left at room temperature. That's 22 degrees Celsius, which is about 70 degrees Fahrenheit, and a relative humidity of about 65%. That's like normal. The inoculated object achieved a desired time point for immediately soaked to elute the virus. So they try to get the virus off the surface. No infectious virus could be recovered from printing and tissue paper after 3 hours, whereas no infectious virus could be detected from treated wood and cloth on day 2. By contrast, SARS-CoV-2 was stable on smooth surfaces. So on glass, it took 4 days before they couldn't identify any virus, and 7 days for stainless steel and plastic. So, different labs have different levels of detectability. So this lab was a little more sensitive. Okay, so it probably was non-infectious, like to an actual person, but 7 days is a long time. So if like someone coughed onto like a door handle, then you could theoretically get viable virus off of it up to a week later. Now, just because that there is viable virus on it doesn't mean that if you touch it, that you'll get it. You'll get COVID by touching a surface. It's probably not because you need quite a bit of viable virus for you to get infected. It's just like one virus isn't going to do it for you. You need probably thousands for it to make it through your innate immune system. And when you touch the door handle, only a small percentage is going to end up on your hand. And then if you wash your hands, that'll get most of that off, and then it won't be a problem. But if you like immediately touch the door handle and then rubbed your nose, then you're delivering a portion of that virus into your nose, and then it has to go through the innate immunity system in your nose, which is like the mucous membranes and the mucus layer. And so you need to have quite a bit of virus before you can get it. Now, if someone sneezes right in your face, you're getting tens of thousands, hundreds of thousands of viral particles, and probably 1 or 2 viruses are going to make it through your defenses and get you infected at that point. But I wouldn't be too worried about the 7-day stainless steel. But if you wanted to know what surfaces are most likely to keep viable virus, well, it's both of these studies show that it's stainless steel and plastic. They can be, depending on how sensitive your assay is, you can detect it up to 7 days afterward, which is obviously quite a long time. And unfortunately, stainless steel and plastic is one of the more common surfaces that we deal with. A lot of countertops are plastic or glass type material, and door handles, the inside of a car, that kind of thing. All right, so now what's the good news? The good news is that there's easy ways to disinfect it. Let's look at this. This is from the Department of Homeland Security. I guess they decided that COVID was a Homeland Security issue, so they wanted to know. They tested a bunch of different disinfectants, surface disinfectants, and figured out which ones were effective and not effective. So if here you see highly effective, partially effective, not effective. So hydrogen peroxide, not effective. So don't think hydrogen peroxide is going to work very well. Then this This bleach with vinegar, not so good. This 70% isopropanol, that's like rubbing alcohol, not so good. But ethanol, which is the drinking alcohol, is pretty good. 70% ethanol. So that would be like, I think some hard liquors are about 70% ethanol. That would work. That's very effective. At 5 minutes of contact will kill the virus. And probably the easiest thing is bleach. So just regular bleach. If you just take regular bleach and do it 1 to 32, 1 part bleach to 32 parts water, that's very effective. Lysol disinfectant spray, also highly effective. And same thing in this study. So anyway, bleach is something we all have probably, and that is a very effective disinfectant for surfaces. Also, naturally, what it is sunlight rapidly inactivates SARS-CoV-2 and most other viruses. Most viruses are not very stable under sunlight. Sunlight has ultraviolet radiation, which is very disruptive to viruses. And so 90% of infectious viruses inactivated every 7 minutes. So when it's so basically the half-life of virus is probably like in the order of like 3 or 4 minutes in sunlight. And this is, they gave the amount of sunlight that you would expect at 40 degrees latitude, which is like kind of like the latitude of, I think that's about the latitude of like Washington, D.C. or something in the summer at sea level on a clear day. So if you're higher up, you get more radiation. So if you're like Denver or something, or Mexico City, you're going to get more solar radiation. So it'll be more effective. If it's winter, it'll be less effective. But overall, sunlight is a very effective way of inactivating the virus. Early on in the pandemic, we all didn't have enough good masks, and all the hospitals were running out of N95 masks. And as physicians and nurses, we were given like 1 or 2 masks and said that we need to make those last for several weeks. And what we would do, what I told my partners and the other people I work with at work to do, is basically take your N95 mask home and set it outside or on your dashboard of your car, and it will, within an hour, it will easily kill all the virus on there. And so we would just switch out the masks after leaving them out in the sun for a day. So one day, one mask would be out in the sun, the next, while we were wearing the other mask, and then we'd kind of switch back and forth. And that's how we made N95 masks last a lot at the beginning of the pandemic. But anyways, so the take-home point is, on plastic and steel, the virus is pretty stable at room temperature in the dark, and you can retrieve viable virus up to about 7 days after inoculation. But that level of virus probably is not enough to really be a problem unless you are like, actively licking the surface and trying to get it as much as possible. But indirect contact, that's probably not enough viral particles. I would say realistically, probably a day or two, which is quite a long time actually for plastic and steel. So that's why good hand hygiene is good. And then if you want to disinfect, Lysol and bleach are very effective. And of course, sunlight is kind of nature's natural disinfectant. So being out in the sun, you're pretty safe from being infected by a virus. All right, so I hope that's helpful for people, and I'll put the links to these studies down in the description. Thanks for watching.